The third part of chapter 4 covers protein synthesis, or making proteins. So we're going to talk about how DNA and RNA is used to make proteins. Let's talk about how DNA is called, also known as deoxyribonucleic acid. It's the genetic material, and it stores instructions on. This is a review from the previous chapter. DNA and RNA are made up of nucleotides. And so here this picture is showing a nucleotide which is made up of phosphate, sugar, and a base. So imagine these stacked on top of each other to make RNA. And if they're stacked on top of each other but you have two strands of them attaching in the middle, then you would call that DNA. The structure of DNA, so it's two nucleotide chains twisted into a double helix. Each nucleotide, once again, is talking about how it has the sugar, which is the deoxyribose, phosphate, and a base molecule. In the middle, the base is attached for DNA. Hydrogen bonds are holding the bases together. A attaches to T, and C attaches to G. This is called a complementary base pairing. Protein synthesis. Here, this slide is important. It's talking about how there are two steps of protein synthesis. There's transcription and translation. Know that transcription really is just making mRNA by using the code in DNA. RNA polymerase is the enzyme that makes mRNA during this step. Then this mRNA is used for translation, during translation. But in addition to mRNA, we're going to see tRNA molecules used and ribosomes to make proteins. And so translation requires the ribosomes. Either the ribosomes could be attached to the rough ER or floating inside the cell. Okay, so step 1, transcription. During this step, the DNA is actually opened up. It's unwound, and one strand of DNA is used to form mRNA. So the picture here shows the pink part. That pink is the RNA polymerase, that's the enzyme that opens up the DNA. It's also DNA helicase here. So using only one strand of DNA, looking at those bases, then the enzyme grabs free RNA nucleotides that are floating in this region. So it grabs the RNA nucleotides and attaches them to the opened up base on the DNA. But here, it needs to be complementary. So if on the DNA there is an A, then the U RNA nucleotide is grabbed by the enzyme and attached to that part. But if there's a C, G is attached. If there's a G on the DNA, then a C is attached for the mRNA. If there's a T, then A is attached. So if there's a DNA, U, A to U. If there's a T in DNA, that turns to A, C to G, G to C. So on the exam, if I give you some letters for the DNA, can you write down or pick the correct choice for the mRNA sequence? So let's do an example. C, A, G, T, G, A. Okay, so what would the sequence be for the mRNA? C is going to turn to G, A to U, G to C, T to A, G to C, A to U. Once the mRNA is made, the DNA gets back to its original double-stranded form. Those bases are going to attach back up, and the mRNA comes out of DNA. Then the mRNA leaves the nucleus. So then we can start the second part of protein synthesis, which is translation. So during translation, converting the genetic code carried by mRNA into a sequence of amino acids. So then the amino acids attach together to become a protein. Translation occurs in the cytoplasm. And know that translation requires ribosomes. Has to happen on the ribosomes. So mRNA leaves the nucleus, binds to the ribosome, acts as a template. Now we're going to see that on the mRNA, each 3-base sequence in mRNA and DNA is called a codon. Then we're going to see tRNAs come over and bring over a specific amino acid. tRNA is just another molecule, it's an RNA molecule. But on the tRNAs, we're going to see that it has 3 bases on the bottom part. Those 3 bases on its bottom is called the anticodon. Okay, so know that tRNAs carry over an amino acid, and it contains the anticodon. This shows what codons then translate into what amino acids. Do not worry about memorizing this, but we do notice here that some codons are for stop, so that's for stopping protein synthesis. And then the AUG is the start codon. Okay, so let's see here how translation is happening. This part here is the smaller piece of the ribosome. And the one with the letters here, this long part, is the mRNA. Each 3-base sequence on mRNA is called a codon. So here AUG is a codon. Then CCG right next to it is another codon. Okay, then at the top here, this is the tRNA. tRNA, or a transfer RNA, has kind of a T shape to it. In the upper part, it carries over an amino acid. And so here it says methionine. Then in its bottom part, it has 3 bases, and that's called the anticodon. So here the anticodon is going to be complementary to the codon. This picture is missing the letter on the anticodon. So let's look at what that letter could be. If the codon for the mRNA is AUG, well, what would be the anticodon on the tRNA that matches to it? So A to U, that matches. So if there's U on the mRNA, then that's going to be, this is supposed to be a UAC on the tRNA's anticodon. And so then the G to C. So only this tRNA is able to bind here. So only if it has the UAC as its anticodon. And then the UAC matches to the methionine amino acid. Okay, so once they get together, so then here the larger part of ribosome also attaches. So here the first tRNA attached, also now the second tRNA is coming in. So here the second tRNA's anticodon in this case is GGC, and so that's the only one that can attach here since the mRNA's codon is CCG. And so C to G, G, and then CG. Okay, but so this one has a different amino acids. And so this is going to continue. And more tRNAs come in, bring over their amino acids. So once on the mRNA a stop codon is encountered, then no more amino acids are going to get added. So all of this is going to disassemble. The ribosome parts are going to come apart from each other. mRNA comes out. So then those amino acids that attach together is then called a polypeptide, or the protein. So this polypeptide can fold into its unique 3-dimensional shape. So with protein synthesis, there are 20 types of amino acids. There are 64 possible codons. Start codon is the AUG, and there are the 3 stop codons. Then a little bit about mutations, changes in genetic information. So 99.9% of our DNA is the same for all humans, but 0.1% is responsible for the differences in our physical appearance. It could be, other organ functions. So these DNA sequences that are different, well, they affect our health, they affect appearance. But some base variations may have no observable effects. So mutation, this one definitely no. Mutation means changes in the DNA sequence. Mutations can occur when bases are changed, added, or deleted. Mutations can happen on their own, they can be spontaneous. So it's spontaneous, this can happen, for example, if insertion of an unstable base occurs into the DNA sequence. Induced mutations, on the other hand, these are caused by mutagens. Mutagens can be chemicals or radiation that cause a mutation. Some mutations are not harmful, do not affect health, but many mutations can have consequences to our health by changing the amino acid sequence. So it's talking about here, for example, Duchenne muscular dystrophy results from a mutation in the gene coding for this protein called dystrophin. So because of this mutation, muscle cells collapse and it results in muscle weakness. But some mutations are actually good. So some people have this mutation that protects them against HIV. So with this mutation, the receptor to which the virus wants to bind to is non-functional. The virus cannot attach to it if this mutation is present. So mutations occur uh, a lot in our cells. Thankfully, we have these special proteins that help to repair these mutations or any mistakes. So we have these special repair enzymes. Now, some mutations are not going to result in any changes because if you noticed, more than one codon may specify the same amino acid. So it says here often 2 to 4 codons specify the same amino acid. Some mutations do not result in production of a different amino acid. Now at the bottom it talks about how having 2 copies of each chromosome is what happens when the egg cell and the sperm cell get together during fertilization. So it talks about here, if one copy is mutated, the other copy may provide enough of the gene's normal function to maintain health.